Hello, ladies and gentlemen. It's my great pleasure to share with you my recent work about calcium study in fruits, especially in lychee. And my topic for this report is dynamics of calcium oxalate in a pedicles of lychee and longan and its relation to oxalate oxidase. First, let me give you some background. You all know that calcium is an important, essential macroelement in plants. And calcium has many functions. And calcium deficiency in fruits cause various disorders, including fruit cracking, bitter pith, blossom rot, and heart rot. Calcium deficiency disorders include cracking type disorder, which is related to low structural calcium in the cell walls of the fruit skin. And the rot type disorder is a result of low membrane bound calcium due to high cell wall bound calcium or low free calcium in the apoplast. And it is generally believed that calcium transport to fruit is through xylem or apoplast pathway. However, calcium uptake via phloem symplastic pathway cannot be ignored. There are evidence showing that phloem symplastic pathway is also important for fruit calcium uptake. For example, calcium is more distributed in the phloem of the pedicel. And gathering, which cuts off the phloem pathway, cause fruit to reduce in size and inhibit fruit calcium uptake significantly. And the strontium is an element which can be regarded as analog of calcium and can be used to trace a calcium movement. And if we feed a strontium to the bearing branch, we can find in the pedicel there is more strontium distributed in the phloem than in the xylem, which is similar to the distribution of calcium in the phloem, as the right picture shows. Therefore, there is a large amount of calcium stays in the phloem. And the large portion of this calcium in the phloem is in the form of calcium oxalate. And in crystals, we can find a lot of crystals around the sieve element in the phloem. Therefore, there is an intensive calcium flux from the xylem to the phloem via calcium channels, which allows the mass flow in the phloem to be an important contributor to the fruit calcium uptake. And the formation of calcium oxalate in the phloem cells help to maintain a low calcium concentration in the phloem sap, allowing continuous mass flow in the sieve element. Calcium oxalate is widely present in plants in different shapes and forms. And the studies have shown that formation of a calcium oxalate crystals is metabolically controlled and reversible. That means calcium oxalate crystals can be dissolved and can serve as calcium storage. The enzyme responsible for dissolving calcium oxalate is oxalate oxidase. And the first gene identified as oxalate oxidase is called germin, first found in wheat. And later, more and more plants were found to have a similar protein called germin-like protein. And some of the germin-like proteins have oxalate oxidase activity. Therefore, germin-like proteins may regulate calcium oxalate formation and thus calcium mobility. In this study, we want to address the following issues. First, dynamics of calcium oxalate in the pedicles of litchi and the longan. Second, we want to look at the relation of relations of oxalate oxidase to calcium oxalate in the pedicles. And three, possible litchi germin-like genes encoding oxalate oxidase. And finally, role of germin-like protein in calcium oxalate formation and food calcium uptake. Here's the materials we used. First, we used 2 cultivars of litchi. One of them, lymphonol, which is cracking resistance with high ability of calcium uptake in the fruit. And second, normaichi, which is cracking susceptible with weak ability of calcium uptake. And third one is Xu-Ming Huang, which is also cracking resistance. We take samples at 6 stages. These are the 6 stages. Stage 5, after stage 4, is ripening stage, in which fruit is undergo rapid expansion. And that is the stage where there is a high demand for calcium. First, we look at calcium content in the fruit and in the pedicles. In a pedicle, we can find that a calcium increase to stage 3 or stage 4, and from stage 4, calcium content decrease as fruit ripes. And comparatively, we can find that normaichi has relatively higher calcium in the pedicle in later stages. And in the pericarp, we can find calcium concentration is generally much lower than calcium in the pedicle. And the normaichi has the lowest calcium content in the pericarp. In contrast, it has the highest calcium content in the pedicle in later stages. Then we look at the calcium oxalate content in the pedicle. We found the calcium oxalate concentration fluctuate with fruit development. And in Xu-Ming and the lymphonol, we can find that its content decrease in stages from stage 4 in later stages. But in normaichi, calcium oxalate tend to accumulate in later stages. Therefore, normaichi has lower ability to remobilize calcium in oxalate. And here we can find calcium crystals in the pedicle. The upper figures shows calcium oxalate in the phloem, and below calcium oxalate crystals in the pith and the xylem. We can find that there are many more crystals in the xylem and the pith in normaichi than in lymphonol and Xu-Ming Huang. Then we measure oxalate oxidase activity, and in the pericarp, the enzyme activity decreases as fruit develops. And generally, normaichi has a weaker enzyme activity in the pericarp. But throughout the experiment, normaichi has the lowest enzyme activity compared with lymphonol and Xu-Ming Huang. And the enzyme activity change pattern is similar between the 2 tissues. We analyze the correlation between oxalate oxidase activity and calcium oxalate content. We just found a weak negative correlation between them. Then we screened germin-like genes from litchi genome. And we identified 5 of the genes, GOP1, GOP3, and GOP2 have premature termination codon. So we focus on germin-like protein 4 and germin-like protein 5. The expression pattern is quite different between the 2 genes. And we can find that GOP4 expression pattern is quite similar to enzyme activity of oxalate oxidase. And correlation analysis show that GOP4 has a stronger positive correlation with the enzyme activity. Therefore, the enzyme might be encoded by GOP4. Then we want to check the role of this gene in calcium oxalate formation and food calcium uptake. Then we silence GOP4 genes with VGAS. And as a result of gene silencing, we can find that oxalate concentration was significantly increased when the gene is silenced in lymph node in the pedicles. And we can find more calcium stays in the pedicles, and higher percentage of calcium in the pedicles is in the form of calcium oxalate, and with less percentage of soluble calcium. As a result of silencing the gene, calcium content in the fruit is also reduced. Therefore, this gene plays a positive role in food calcium uptake. Here's our conclusion. Calcium oxalate level in the pedicles fluctuate with fruit development, suggesting its formation is reversible and the calcium oxalate can be reused during fruit ripening. And a normal tree with weak calcium uptake ability has weak activity in remobilizing calcium in calcium oxalate. And second, oxalate oxidase might prevent calcium oxalate accumulation in the pedicles. 3, GOP4 is very likely the gene encoding oxalate oxidase in lychee. And 4, GOP4 regulates calcium oxalate accumulation in the pedicles and plays a positive role in food calcium uptake. Finally, I want to acknowledge fund resources supporting this study, and also, very importantly, students who contributed to the study, including Dr. Yijing Wen, Dr. Song Wenpei, Mr. Chen Wei, and Dr. Ferry Ott. Thank you very much for attention.